This is OwlUp, where learning happens in minutes, not hours. For a deeper journey, explore all our courses at owlup.co. Now, let's dive in. Welcome back to Chapter 2 of our series, Evolutionary Oddities. In today's episode, titled The Lazarus Species, Creatures That Cheat Death, we'll explore the incredible adaptations that allow certain species to survive extreme conditions that would spell doom for most. So, let's dive in. A frog's heart stops beating, its blood turns to ice, and scientists still don't call it dead. In another lab, a dust speck animal dries out for years, then springs back to life with a drop of water. Today's question: where does life actually end if some creatures can press pause? Biologists now group these survival stunts under one broad banner: cryptobiosis, life so slowed that instruments can barely detect it, yet not quite gone. Instead of fighting lethal cold or drought in real time, these organisms quietly exit the game. They stockpile unusual molecules, rearrange their cell interiors, and let almost everything grind down to a whisper. Some even swap liquid cellular contents for a glassy state that locks structures in place, the way cooled caramel hardens on an apple. The result isn't immortality, but an extreme hedge against bad luck. Frozen forests, dried up ponds, even outer space become less like death sentences and more like long, risky layovers. And when conditions finally shift in their favor, chemistry reverses, motion returns, and the question becomes stranger. Not just how did they survive, but what exactly was surviving all that time? Some of the most striking examples come from places that look utterly lifeless: salt-crusted lakes, polar deserts, even ancient permafrost. Under those hostile surfaces, Lazarus species wait, not for minutes, but for decades or longer. Wood frogs time their frozen stasis to winter, then thaw in step with the forest's melt. Tardigrades and brine shrimp, by contrast, ride out unpredictable disasters: drought, radiation, even vacuum. Their trick isn't just endurance, but flexibility. Evolution has turned them into biological savings accounts, conserving just enough viable structure to cash out when the environment finally pays off again. Start with the extremes, and a pattern appears. Wood frogs aren't the only vertebrates flirting with the edge of viability. Painted turtle hatchlings can sit in frozen soil for weeks, brains quiet, cells braced against ice. The common porwheel, a small North American bird, drops into torpor so deep in winter that for days it looks, and almost measures, like a corpse tucked into a rock crevice. These aren't accidents, they're repeatable life strategies, tuned by natural selection to match reliably harsh seasons. On a different scale, tardigrades, brine shrimp, rotifers, and nematodes use cryptobiosis not just as a shield, but as a time machine. Their off state lets genes move through years of drought or cold until a lucky rainstorm or thaw finally reveals which lineages made the right bets. Brine shrimp in salt lakes, for example, hedge across generations. Some embryos hatch right away, others remain sealed in hardy cysts that may not reawaken for decades. A single population is effectively spread out over time, not just space. This strategy reshapes evolution itself. When an environment swings wildly, epic droughts, freeze, thaw whiplash, most organisms must adapt on the fly or vanish. Cryptobiotic species can sit out bad years with minimal change, then re-enter the gene pool. That slows the pace at which they need to evolve new defenses, yet it also creates a hidden archive of older genotypes. The permafrost nematodes that revived after tens of thousands of years weren't just a curiosity. They were a snapshot of ancient biology, suddenly reinserted into a modern world. Biochemists have seized on these tricks. Trehalose, the sugar many of these organisms rely on, now helps stabilize vaccines, enzymes, and even human cells for shipping. One lab famously stored yeast at room temperature for over a decade using trehalose-rich preparations, recovering viable cells long after normal samples would have decayed. Space agencies test brine shrimp cysts and tardigrades on orbital platforms, probing how much vacuum, radiation, and temperature swing a living system can truly tolerate. Your challenge this week: pick a perishable thing you normally refrigerate, herbs, bread, leftover rice, and test how small changes in storage, drier, colder, more sealed, partially frozen, alter its lifespan. Treat it as a micro-experiment in survival strategies. By the end, ask yourself, if a plant leaf or loaf can last longer with simple tweaks, what might evolution accomplish over millions of years of tuning the pause button on life? In finance, investors spread money across low and high-risk assets to ride out market crashes. Many Lazarus organisms do something similar with time instead of cash. Tiny crustaceans in temporary ponds produce eggs with staggered wake-up schedules. Some hatch after the first rain, others after several wet-dry cycles, and a fraction may wait years. Each batch is like a different maturity date on a bond portfolio, reducing the chance that every generation emerges during a catastrophe. Soil microanimals layer their bets vertically as well as temporally. Those deeper down may never experience the same droughts, freezes, or heat spikes as their surface cousins. Even plants join in. Seeds from a single desert plant can germinate over multiple seasons, ensuring that one freak year of heat or herbivores doesn't erase the lineage. Cryptobiotic phases aren't just emergency shelters, they're built into long-term reproductive planning, quietly shaping which genes get to see the next good year and which remain archived. Life that can pause itself forces a rewrite of our risk maps. Deep freezers of soil, salt, and ice may quietly store lineages and their microbes far outside normal quarantine nets. Drug developers already borrow their tricks, but urban planners and conservationists might have to as well. Dormant seed banks under parking lots, spores in dried fountains, even lab contaminants that wake after a power outage could matter in a warming, flooding, rapidly disturbed world. We may be treating dead and alive like a light switch when nature uses a dimmer. As we learn to borrow these pause tricks, stabilizing donor organs, preserving endangered embryos, maybe even backing up human cells like photos in the cloud, we're quietly expanding what a lifetime can mean and who or what gets a second chance at it. Here's your challenge this week. Pick one Lazarus species from the episode, like the coelacanth, the Wollemi pine, or the Lord Howe Island stick insect, and spend 20 minutes tracking down a real conservation project currently working with that species or its habitat. Once you've found one, commit a specific action in support. Either donate a fixed amount, even $5, sign up for their newsletter, or register for one upcoming event/webinar they run. Before the week ends, share the species Back from the Dead story plus the project's link with at least one person in a message, post, or email, and explicitly ask them to support it in the same way you did. This was OwlUp, your daily step forward. To keep learning with structured courses and more insights, go to owlup.co. Until tomorrow!